I'm Pribyl. I'm a postdoc at MIT and I'm a Superfund trainee. Everyone is exposed to chemicals through their drinking water and diets every day. One chemical of concern is N-nitrosodimethylamine, or NDMA, which is present at high levels at the Olin Chemical Superfund site in Wilmington, Massachusetts. In the 1990s, there was a childhood cancer cluster discovered in Wilmington. Later, a study was done and it showed that there was an association between the women who were exposed to NDMA during pregnancy and cancer in about 22 of their children. More recently, NDMA has been found in over a dozen commonly used drugs, which have since been recalled, including blood pressure, diabetes, and heartburn medications. While exposure to certain contaminated drugs was a significant source of NDMA exposure, we all actually consume small amounts of this chemical every day through our diets. And these include processed foods like cured meats, bacon, fish, cheese, some vegetables, and malted beverages like beer and bourbon. Exposure to high concentrations of NDMA in lab animals has proven that it can cause cancer. For this reason, it's considered a probable human carcinogen. Our group at MIT studies this particular chemical because there are a lot of things about exposure to NDMA that we simply still don't understand. So we know that we have a couple of very important genes and proteins in our bodies that help break down NDMA and usually protect us from its harmful effects. However, we don't really know if you're lacking one of these proteins, how that may affect your risk to exposure of NDMA and how that could maybe lead to increased odds of getting certain types of cancer. Because we know what these genes and proteins are, we can use living organisms like mice that share many genetic biological features that humans have to model or study the effects of exposure to this chemical. Further, we can take advantage of the fact that we can use mice that lack these protective genes so that when we expose them to a chemical like NDMA, we can then monitor them over time to see exactly how they develop cancer. One of the unique aspects of our mouse model is that it has a tiny green fluorescent protein inside each of its cells. And this acts like a light switch. If there is a mutation in the DNA of a cell, then the light is on. But if there is no mutation, then the light is off. We can then track the number of lights that are on to see how and when mutations are appearing, and in which cells or organs of the mice to see how fast or slow mutations arise after exposure to NDMA. This type of technology could help us not only predict the short and long-term effects from exposure to NDMA, but possibly many other chemicals. The bad news is that we are all exposed to various chemicals over our lifetimes, whether we choose to or not, through our diets and environment. But the good news is that we are coming closer in our ability to more accurately predict what effect these exposures may or may not have on our bodies. And that is why this type of research is so important for all of us.